Hello, welcome to Chapter 2, titled Heredity, Prenatal Development and Birth. Let's just jump straight into it. All right, so let's get some basic definitions. Starting with the genetic code. The sperm cell and an ovum each contribute 23 chromosomes to the zygote for a total of 46 chromosomes. Sperm and ovum are produced by meiosis. Chromosomes are slender threads inside the cell's nucleus that carry genes, the genetic material. And the DNA is a double helix-shaped molecule that stores everything cells need to replicate. So very definitionally based right now. So there's actually a federally funded project called the Human Genome Project. The goal of this project was to map the sequence of chemical units / letters that make up the strands of DNA in a full set of 46 human chromosomes. 3% of the human genome consists of what was defined as genes, parts of the DNA that are transported into RNA acts as templates for protein production. Other 97% play critical roles in regulating activity of the protein-producing genes. 99.9% of the genome is the same across all humans. So basically in English what this found was that genetically humans are almost nearly identical. We're talking about 0.1% difference among genes across humans. And only 3% of the human genome consists of what we typically think of as genes. And there's a video in the previous slide that you can check out. Moving on to cell replication. Genes are specific sequences of nucleotides and are recipes for making proteins. Proteins are responsible for influencing the structure and function of cells. So again, this is going to be kind of a definition-filled PowerPoint slide, just like kind of the nature of the bio chapter. Mitosis, cell divides to produce two identical cells, each containing the same 46 chromosomes. Zygotes become multiple cell organisms through mitosis. In meiosis, gametes chromosome duplicate, divide twice, resulting in 4 cells containing only half the genetic material of the original gamete. And I think the figure on the right kind of illustrates this well. You see at the top mitosis producing identical cells for all cells except for those used in reproduction. So literally just making exact copies. And the meiosis you see below it, taking two separate cells and then combining 50% with each. So pretty straightforward. Now let's look at twins. We have identical twins and fraternal twins. Identical twins, easy way to go about this is just saying MZ. They originate from one zygote. It's one fertilized ovum divides to form two identical individuals. There's two, obviously. One out of 250 births are identical twins. Fraternal twins, again, DZ, simple way to get around having to say the word, is a result from two ova, and they were released at approximately the same time, and each is fertilized by a different sperm. This occurs 1 out of 125 births. So one splitting in half, basically, or sorry, I should say one fertilized ovum divides to form identical individuals versus two ovums. And so that's why you can have fraternal and identical twins. So looking at sex chromosomes, chromosomes of the 23rd paired are the sex chromosomes. Males have an X chromosome and a Y chromosome. Females have two X chromosomes. And the father determines the child gender. The sperm carries either an X or Y chromosome. And to the right, you can see a figure of the male and female chromosomes. Genotype versus phenotype. Genotype is our genetic makeup. A phenotype is our observable trait. Gene expression is the activation or deactivation of genes by environmental experiences throughout development. So what we're really talking about here is just because you have a particular gene does not mean the gene will be expressed. And that's what we're talking for gene expression. When the gene is expressed and is observable in the real world, that is referred to as a phenotype. So you can have, for example, a genetic predisposition for behavioral issues, but you need the environment to activate genes that allow the genes to be expressed, and then they're observable, thus their phenotype. So there's a difference between having a predisposition for a gene and having the expression of a gene, which is typically triggered by an environmental event. And we'll get into more of this later. Single gene pair inheritance. Thousands of human characteristics are only influenced by only one pair of genes, one from the mother and one from the father. There's a dominant gene, a gene that will be expressed when paired with a recessive gene, and a recessive gene, which is a weaker gene that can be dominated. And I pulled this chart to the right from another textbook, but this is kind of what we're all taught in basic biology, that brown eyes are dominant and eye colors such as blue and green, for example, are recessive traits. Many and most psychological traits, in contrast, probably aren't influenced simply by dominant or recessive genes. They're influenced by hundreds or thousands of genes, each exerting tiny effects. And that's one of the big takeaways I want you to have from this chapter, is that there's really not single individual genes that express behavior. So there's not a gene for alcoholism, for example. Rather, genes interact with each other, as that definition or that bullet point says, dynamically. The environments affect the expressions of genes, and it's really not just one gene, it's thousands of genes, networks of genes. It's actually kind of similar to more commonly understood brain networks. It's not just one individual part of the brain, but collective networks working together. And it's a similar principle with genes. Chromosomal abnormalities result when a child receives too many, too few, or abnormal chromosomes at conception. Down syndromes, Turner syndrome, XYY syndromes, main cause of pregnancy loss. So pretty straightforward. You receive too many or too few chromosomes, and then you're at risk for neonatal death. Mutations. So this is another way that genes are changed. Mutations are a change in the structure or arrangement of one or more genes. Can be beneficial depending on the environment and type of mutations. Odds that mutations will occur are increased by radiation and toxic stress. Most are just spontaneous errors during cell division. Odds of mutations increase as parents get older, typically a different version of a particular gene. So the idea here is that naturally within the environment, through just natural selection or errors within the cell replication process, there will be mutations of genes. Some of them will survive, some of them will die, and organisms will die as a result. And we know that the environment also impacts the mutation of genes. So for example, Chernobyl, we know that animals that live in that area have different genetic genotypes and phenotypes than members of their exact same species living in non-nuclear waste polluted areas. Genetic diseases and testing. So there are one way to test for genetic diseases during pregnancy and genetic counseling, which provides information about nature, likelihood, and effects, and treatment of genetically based diseases and disorders. Carriers do not have the disease, but can transmit the gene for it to their children. So sickle cell trait versus disease, for example. So sickle cell trait is found in humans across anywhere where there's mosquitoes. So the Mediterranean, Africa, Middle East, and they actually protect against partial protection, they provide partial protection against a type of malaria. So on the one hand, that genetic trait actually protects an individual from a predator, mosquito, a threat to their well-being in their environment. However, when environmental insults, such as low oxygen environments, their red blood cells deform into a sickle shape and become adhesive, sabotaging the cell's ability to carry sufficient oxygen and causing them to block small blood vessels, including capillaries. These events trigger excruciating painful episodes. So the idea here is just because you have this sickle cell trait does not mean that you have sickle cell disease. And you can see that the environment actually impacts the progression of from turning a sickle cell trait to sickle cell disease, and that environmental impact being low oxygen environments, which place individuals with this trait at risk for painful episodes. Behavioral genetics is a scientific study of the extent to which genetic and environmental differences among people are responsible for differences. Heritability is the percentage of the variability in a trait across individuals that is due to genes. Some traits are highly heritable, such as height, while others are not, religious affiliation. Height among plants may be largely heritable, but however, watering these plants and environmental manipulation can result in substantial increases in their heights. High heritability doesn't imply unchangeability. What I found interesting about this textbook, it points out that it's impossible to say what scores on any psychological test is percentage from genetics, and then many times in the text they throw out estimates of whatever the psychological construct is, personality, IQ, whatever, is 40% based on genetics. So I've found that a funny contradiction. But in general, I would agree that it's not possible to definitively know and precisely know exactly what percent a psychological construct or a phenotype is caused by genes for a lot of reasons I will get into momentarily. And even in traits like height, for a plant example, they if you don't water your plants, they're going to die. We found that out in quarantine quite frequently. And even among humans, my research in the Democratic Republic of the Congo found that maternal stress during pregnancy, those children born to, were fetus during high levels of maternal stress, specifically military occupation, they were born shorter than children who were not exposed to that military occupation. So you can see, even with highly heritable traits like height, the environment still impacts that phenotype. Okay, continuing on behavioral genetics, scientists use three types of designs to estimate heritability in traits: family studies, twin studies, and adoption studies. So starting with family studies, what characteristics run in the family? Biological relatives share environmental and genetic material that are the same. So you live in the same house, and you're related to X amount of percentage to your brother or sister. Moving on to twin studies, we have MZ and DC, which we just reviewed. And looking at DZ specifically, they share about 50% of genes on average, no more genetically alike than ordinary brothers and sisters. If identical twins are more alike on a psychological characteristic than fraternal twins, researchers can infer the psychological outcome is more genetically influenced. So the idea here is, if identical twins have an outcome that is more similar, a psychological outcome that is more similar. So, if their self-esteem is more similar than fraternal twins, we can infer, or these researchers argue, you can infer that the reason why those identical twins, are more alike on self-esteem is because they are genetically, 100% of their genes are the same. So you're kind of comparing and contrasting. You're saying, oh, look, these identical twins are 100% the same on this self-esteem measure. Well, these fraternal twins are, just make up a number 50% the same. Therefore, 50% of the self-esteem is derived from genes. That's kind of a simplification, but that's kind of the logic behind it. In adoption studies, children adopted into new homes resemble their adoptive or biological parents. Share genes, but not environment can assume genetic influence. So the idea here is, if you have identical twins, again, sharing 100% of the same genes, but say they're adopted, one's adopted to a family in Australia, one's adopted to a family in Bolivia. If they turn out that they have the exact same self-esteem levels, based on whatever, some self-esteem test, then these researchers argue that we can infer that self-esteem is, whatever percentage from genetics. One issue with that, though, is selective placement. Adoption agencies place children in similar homes as biological parents. So the idea here is that, say your family, is X race, X class, whatever. Adoption agencies try and reduce the stress on children by placing the children into homes that are as similar as possible as their original homes. So that begs, the question is what driving the differences between identical twins and fraternal twins, the actually genetically based, or is it the environment because the original biological parents and home environment is being replicated now in their new adopted family's home, by design through selective placement. Moving on to genes in the environment, genotype environment correlation specifically, which is a process by which genetic factors contribute to variations in the environment. People with different genes have different experiences. There are two types, passive and evocative. Passive occurs when children passively inherit the genes in the environments their family provides. Because parents provide children with both their genes and a home environment compatible with those genes, the home environment to which children are exposed are correlated with their genotypes. So the idea here is we know that children inherit 50% of their genes from their father and 50% of their genes from their mother. So, but those people, just don't disappear from their life. They are being brought home, and live in, and especially in early years of their life, a home environment created by those exact same parents that gave them those genes. So the parents are creating home environments that are reflective of those genes. So children are passively taking in that home environment, which is partially or fully um, designed by people with certain genetic archetypes, which again are 50% in the case of the infant, 50% from the mother and father. Now, moving on to evocative, how the social environment reacts to individuals based on their inherited characteristics. Whether one has a more outgoing or shy temperament will affect how he or she is treated by others. So the idea here is that if a child is born with a very argumentative temperament, for example, well, people are probably going to be argumentative back to them, and or hostile towards them, which then reinforces that gene, and then it's kind of a cycle where their genes are being reinforced by their environment. But the important part of this is that it's the individual acting on the environment that reinforces their genotype. So passive is passively hitting you. It's your home environment from typically your parents, and evocative is a child acting on the environment, and because they act in a certain way based on their genotype, they receive reinforcement back from the environment, which reinforces their original genotype. Active genotype environmental correlation occurs when individuals seek out environments to support their genetic tendencies. The individual with a genetic predisposition to be inverted is likely to go out every night, stimulating social things. Okay, so kind of almost exactly the same thing here as evocative. But in this case here, individuals are seeking out environments that support their genetic tendencies. And that's how I want you to conceptualize this different for the purposes of this class. You are seeking out environments. So just as the sample says, say I'm extroverted, and so then I'm going to go out and I'm going to talk to people, and then it's presumably people like me, and I feel great about it. Well, that reinforces my genetic predisposition of extroversion. And so then I go out again, and then bam, 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 bam. Or, that's just like the active, just like the evocative, but in this case, active, I'm seeking out an environment that supports my genetic tendencies. And then genotype environment interactions involve genetic susceptibility to the environment. The concept of gene environment interaction tells us that people with different genes react differently to the experiences they have. By contrast, the concept of gene environment correlation says that people with different genes have different experiences. Adoptions studies provide evidence for genotype environmental interactions. So the idea here is that the concept of gene environment tells us that if I have a genetic predisposition towards the depression, and I grow up in an environment that's very tough, whatever. I grew up in poverty. I grew up with a lot of family violence, something along those lines. I am going to react to that poverty, that family violence, differently than someone who does not have that genetic predisposition for depression. Namely, I'm more likely to get depressed due to my environmental circumstances. So it's the same environmental circumstances, but my genetic predisposition, my genetic susceptibility to an adverse environment makes me more at risk for getting depression. So it's different. So the difference again is gene environment interaction is people react differently to the experiences they have versus gene environment correlation, which tells you that people have different experiences. So same experience, reacting differently versus different experiences. Epigenetics examines how environmental influences affect the expression of genes, process through which nature and nature co-act to bring forth particular developmental outcomes. Might explain differences in identical twins. So the idea here is, and you'll see the video in the notes, that the environment actually impacts the genetic expression, that is, again, your phenotype of an individual. So just because I'm genetically a genetic predisposition towards depression does not mean that I'm going to be depressed. However, if I'm in an adverse environment, I am more likely to become depressed because the environment is literally impacting the genetic expression of my, just hypothetically, my depression gene. That's not how it works, but it's activating my depression gene. It's like a on and off switch, kind of, you can imagine it as. And this might explain differences in identical twins. Because a twin may interact with even though both twins have the same susceptibility towards depression, one twin, might be closer with their parents or might have different social groups at school. So they have different environmental influences that impact the same genetic disposition differently because they never experience it. So you're, among one twin is among or hangs out with a group of drug addicts. The other twin hangs out with a group of nerds. The one who hangs out with the group of drug addicts is more likely to do drugs because their environment is literally activating their genetic predisposition towards addiction, while the nerd is not interacting with that environment, not experiencing that environment. So the environment doesn't have that opportunity to turn that addiction gene on or off, figuratively. Prenatal stages. Embryologists study early growth and development. Prenatal development is broken down into 3 stages or periods: germinal, embryonic, and fetal period. So looking at the germinal period, it begins during the first trimester and it lasts approximately 2 weeks. The zygote divides many times through mitosis, forming the blastocyst, which is a hollow ball about 150 cells that is the size of the head of a pin. Between 8 and 30% of pregnancies end in miscarriages, perhaps a biological method to eliminate embryos who would not survive. So the idea is through natural selection, the embryos which would not make it, I have no chance of surviving postnatally. They abort, and it's as a biological method by which our species survives, by aborting infants that would immediately die and just waste resources, biological resources, for the mother, father, etc. The embryonic period, which is the 3rd through the 8th week after conception. Placenta is a tissue fed by blood vessels from the mother and connected to the embryo by the umbilical cord. The embryo receives oxygen and nutrients from the mother. The placental barrier offers partial protection from potentially dangerous hormones and chemicals. So I'm going to cover this a lot in class, but the idea with the placental barrier is that, say a mother is stressed, then the mother will release stress hormones. Well, the placental barrier is trying to block those stress hormones from reaching the fetus. So there's, kind of a biological barrier that tries to block those hormones, those chemicals from reaching the fetus and damaging the fetus developmentally. And the idea is though that the placenta barrier is, blocks some percentage of hormones, but they don't block everything. So duration and the strength, the magnitude of the hormones and chemicals impact the degree to which they can cross the placental barrier. Now moving on to neural development, organogenesis, the blastocyst differentiates and specializes into special tissues and organ systems, kidneys, lungs. So organogenesis, literally just the genesis, the creations of organs through this process. That's just the term for it. The brain begins forming 3 to 4 weeks, emerges from the neural plate. Primitive parts of the brain develop earliest. So the idea is, the fetus isn't doing calculus. They're not worried about doing that first. It's worried about breathing, sleeping, moving, digesting, and thus those are what develop first. Those areas of the brains that are associated with those activities. And the fetal period lasts from 9th week of pregnancy until the birth. So it's first to the 3rd trimester, technically. Critical period for 3 processes of brain development. That's proliferation, migration, and differentiation. And we're going to walk through all 3. Proliferation. Number of neurons increases by hundreds of thousands every minute during pregnancy. So like this super fast period of neurons forming within a fetus. A concentrated period of proliferation occurs between the 6th and 17th weeks after conception, and infants have around 100 billion neurons. And the proliferation of glial cells occurs after birth. So after birth, we see more glial cells forming, which provide support for an infant's neurons. So the idea here, proliferation, just neurons proliferating, being created. Migration, neurons move from the center of the brain to specific locations where they become parts of larger neural units. They're influenced by biological instructions in the biochemical environment. Some neurons migrate passively, others actively. Once it's at its destination, the neurons begin communicating with surrounding neurons. So migration, pretty straightforward, movement, moving from one place to another. And because each of these neurons have genetic instructions, they know where to go. And then once they get there, they start forming connections with other neurons, because it's not just pockets of neurons, it's the communication. These brain networks begin to develop. Next, differentiation. 2 major theories of neuron specialization, and that's what we're referring to here. Is neurons specialized based on where they land? So what each undifferentiated neuron specializes in depends on where it migrates. First is cells know what they're supposed to be and where they're intended to go on their path before reaching the destination. So the idea here is, how much do the neurons know what they're doing? Are they just kind of floating along to a specific area, and once they get there, the other neurons are like, hey, build some motor movement or something along those lines? Or do they already know where they're going to be? So, the motor neuron cells / neurons are like, oh, I'm going to go to the motor cortex because that's what I do. And myelin. Coat neurons in myelin sheet, which makes neurons electrical signals travel fast. And we mentioned those actually in chapter 1. And the idea here is it's not just the number of neurons. It's really the efficiency and the speed that really matters for these advanced neural networks that develop not just prenatally, but across the lifespan. And in different parts of the lifespan, you see sudden spurts of myelination. Fetal programming. So this is actually one of my specializations, and I'll talk about this, or I'll refer to this kind of a lot throughout the course. And the idea here is that environmental events / maternal conditions during pregnancy alters the prenatal development of the embryo / fetus and sets certain physiological functions. Prenatal experiences can change a person's physiology and brain development. So in English, all that means is that what happens prenatally impacts the fetus. Kind of straightforward, but it was not accepted or is difficult to study for a very long time. But now we know that prenatal experiences matter. They impact lifelong developmental outcomes, depending on the specific outcome. And the fetal programming hypothesis argues that the environment in utero can alter development of the fetus with a permanent effect on the phenotype, affects early life and long-term health, social, and cognitive developments. So again, straightforward. This is just the term for it, the fetal programming hypothesis. And that's, again, that what happens in utero matters. I know, very breaking news for most people. Now, looking at teratogens, any disease, drug, or other environmental agent that can cause harm or can harm a developing fetus. There are sensitive periods, and that's when organ systems are most at risk during periods of rapid development. So you can see on the right-hand side here, this is a standard chart. This is in every developmental textbook. But basically the idea here is, as I was saying before, that certain parts of the brain slash fetus develop at different times due to prioritization, and that's just, genetically, that's how that occurs. Well, because certain organs or parts of the brain or, limbs, whatever, occur, the development rapidly occurs at a particular part of pregnancy. During that rapid time of growth, so whatever for eyes, whatever, just say eyes, I'm just making up something. Eyes happen within the fourth week. Rapid development of the eyes occur within the fourth week. If some big environmental stressor happens on that fourth week, that means that the fetus's eyes would be at an increased risk of developmental abnormalities because they're in a moment of a sensitive period, aka when an organ is rapidly developing and that organ is, or that part of the brain is very in tune to the environment around it. Doses and duration, greater and longer exposure, the more likely serious damage will occur. So pretty straightforward here. The longer a teratogen is present and the higher amount of it that is present, the more dangerous it is for a developing fetus. Genetic makeup, susceptibility to harm influenced by genetic makeup of child and mother. So some fetuses are genetically at risk for, being born blind, or something along those lines, whatever it is. So if a stressor occurs during, again, that fourth week, where I'm just making up that eyes are developing, then the child who has a genetic susceptibility to eye problems are more at risk for that environment negatively impacting their eye development, as opposed to a child who does not have the genetic susceptibility towards blindness. Environment effects depend on the quality of the prenatal and postnatal environments. So this is, again, kind of straightforward. Not only is it that the prenatal environment impacting development, but the postnatal environment affects development. So in these postnatal and prenatal developmental tracts interact in complex ways. 15% of newborns have minor problems, 3% have severe anomalies due to teratogens. So some of the common teratogens: tobacco, rates of smoking decline across pregnancy, mothers who are low income, young, less educated, indigenous, and white women are most at risk. The effects of smoking during pregnancy vary, but it's generally associated with increased risk of miscarriage, respiratory problems, low birth weight, shorter as adults, and poor school performance. Postnatally, children are at higher risk of SIDS, which is when a sleeping baby stops breathing and dies. Now, interestingly, stress is associated with smoking. So again, you see this environmental influence impacting the likelihood of a fetus encountering tobacco, because maternal stress, mother having stress from, whatever it is, poverty, dislocation, in case of an indigenous person, loss of culture, and poverty as well, making a mother more susceptible to kind of self-medicating, which could impact the fetus. Now, one interesting thing about smoking, and it really speaks to the how important the prenatal development or prenatal environment is, that mothers who are undergoing fetal fertilization, so say you a sperm and an egg are fertilized in a lab and then implanted in a different mother, if that different mother smokes, that fetus is still more likely to have low birth weight. So that, it demonstrates that it's not just this genetic predisposition or this genetic destiny kind of that impacts developmental outcomes within the fetus. It's literally no genetic similarity between the mother who has a, a sperm, an egg fertilized within a lab implanted within her. Um, so having no genetic similarity, but yet her decision to smoke still impacts that fetus's development. Now looking at alcohol, it easily passes through the placental barrier, disrupts neuronal migration. So like I said before, the placental barrier, pretty solid, but has a lot of limitations. And specifically for alcohol, it easily passes through the placental barrier. Can lead to neuronal death and impact of glial cells, which is support and nourish neurons, as I previously stated, and can lead to fetal alcohol syndrome, which is facial abnormalities, born lighter, poor growth, and the central nervous damage, um, kind of globally. So you have this hyperactivity, seizures, tremors, lower IQ scores, etc. And it's difficult to entangle the effects of alcohol consumption during pregnancy with other environmental effects. And you'll see this for a lot of stuff in this book. It's not that none of these behaviors occur within a vacuum. A mother who is so desperate, in so much pain that she's consuming a lot of alcohol during pregnancy, guess what? She's a lot of other things going on in her life. It's not just alcohol that's impacting that fetus, and it's not just alcohol is not the only stressor in her life. It's, whatever it is, a collection of things that drive somebody to do that. So trying to disentangle specifically what environmental factor is impacting development is very difficult, and that kind of, you know, calls back to Bronfenbrenner, that we are interacting with all these factors in our family, in politics, and economics, in our neighborhoods, in our states that impact us dynamically. And it's kind of difficult to pinpoint exactly which individual stressor is harming a particular fetus. And so one example of this co-occurring incidence of stressors and the impact of early life stress on developmental outcomes is from this study here. And I'm going to introduce you to adverse child outcomes, or events, I should say, and typically called ACEs, and that's talked about a lot in this book, but I'm going to bring it up a little earlier because I think it's relevant right now. And there are different ways that ACEs are measured, but in this study that we're looking at, it had 11 questions that assess 8 areas of abuse and household dysfunction before 18 years of old. So it's these early life stressors. And the idea here is all of these stressors, like I said before, co-occur. It's not just that, you're abusing alcohol. You've got other things going on in your life. So what we want to do, if I'm an ACE researcher, is to collect the totality of stressors. So measures such as physical abuse, verbal abuse, sexual abuse, and measures of household dysfunction include something like living with someone who abused substances, incarcerating of a family member, witnessing domestic violence. And the idea here is that the collection, the cumulative effect of these ACEs are what matters. It's not one individual stressor, it's the totality of a stressor. And that's the way you should look at the impact of pterogens, of stressors, because it's not only because it's theoretically impossible to disentangle these stressors, but it goes back to that duration and dosage effects. It's the more frequently something occurs, and the more the higher total amount of stresses that occur is what impact really harms development, is the idea here. And you see this in this study here. I just copy and pasted a study that looked at ACEs. And again, the measures are on the right hand column. 6% of participants reported drinking during pregnancy after controlling for a bunch of things. it found that increasing ACEs were positively associated with higher odds of alcohol use during pregnancy. So literally stresses before 18 years old was associated with alcohol use during pregnancy. So again, this idea that this accumulation of stressors not only harm an individual short term, but impact and harm an individual long term and has intergenerational effects. So those ACEs are literally impacting how a mother acts during pregnancy. And I should say as well as fathers and family members. These all occur dynamically within a household. Now, children of cocaine. Now, this is an interesting teratogen that I'm always interested in because there's a lot of history that goes into this. So the idea with cocaine is unclear whether offspring developmental outcomes are because of cocaine use or prenatal and postnatal environments. So cocaine users are less attentive to children, fewer interactions with children. So again, very difficult to disentangle. What is the exact impact of this drug? Is it just a drug, or is it that postnatally these parent-child interactions are less fruitful, they're less positive, the child is more neglected? It's maybe that's what's actually driving a negative impact of cocaine use on a mother and father's offspring. Crack baby hysteria. September 1985, the New England Journal of Medicine published research that described findings that babies born to cocaine using mothers remained smaller, sicker, moodier, and less social. Big issue with this is that there was no control group. They didn't have a control group of mothers who had experienced similar stresses, aka similar ACEs, but did not use cocaine use. So it was, that's kind of a problem with trying to make a conclusion about the impact of cocaine use during pregnancy. And it was a small sample with an N of only 23. It failed to consider pertinent environmental factors such as poor nutrition, housing conditions, and the death of quality prenatal care among mothers. So we understand that the poorer you are, the more stressed you are, the less prenatal care you are. So is that what's actually driving it, or was it the cocaine use? Was it the housing conditions? Was it lead on paint? I don't know. It wasn't controlled for. But regardless of the scientific limitations of this study, and to credit, the author did originally state that these limitations exist. There was kind of a media hysteria about it, and it became a countrywide hysteria. And you can see this: children of cocaine, the inter-crack epidemic is now giving birth to the newest horror, a biological underclass, a generation of physically damaged cocaine babies whose biological inferiority is stamped at birth. So look at the assumptions built on this. You have, obviously, spoiler, this is going to be talking largely about Black children, Black mothers, and we are defining these children as a biological underclass that are stamped at birth, aka they are destined to live a certain life. Well, that's not how, we've just gone over, that's not how genes and prenatal stressors work. There's these environments interact with these babies, all babies, very dynamically, and outcomes are not predestined. If a child who has a genetic predisposition towards something does not encounter a very stressful environment, then that gene won't be expressed. Now I wanted to provide some other examples, and this is from actually a book called Medical Apartheid, if you're interested in. And so just kind of going through this, provided medical support for putative legislation and dehumanization of children. Now I've talked about during our syllabus the impact of our understanding of psychology as a country, how that impacts our political systems. Now here's a big example here. If I believe that a certain drug use or certain behavior by a mom is going to make them a biological underclass for life, so almost subhuman, that's going to make me react politically in a certain way. So there's all kinds of articles I could have pasted here, but Washington Post warned of a time bomb of cocaine babies. St. Louis Post-Dispatch, disaster in the making, crack babies start to grow up. San Diego Union, drug babies invade school. So these biological underclass of predators coming to regular schools and putting our regular, our non-crack babies at risk. So again, there's a lot of implications here for this understanding of prenatal development and postnatal development. In 1997, a group called CRACK, Children Requiring a Caring Community, papered inner city billboards with offers of $200 in cash for cocaine addicted men and women willing to undergo sterilization. 60%, so we don't have exact numbers, but 60% of the recipients were Black or Hispanic. So again, now you have individuals believing that that there's crack baby epidemic, and they're dangerous not only to themselves but to other people around them. So now we're trying to prevent people through, are so stressed out that they are, either addicted to cocaine. We don't even know in that case, but we'll just say they're addicted to cocaine. Instead of helping them, we're trying to sterilize them. Although exposure to cocaine in utero can damage a fetus, a baby cannot be born addicted to cocaine. There is no difference between prenatal exposure to cocaine and to crack cocaine, and some studies have found no difference on IQ tests between children who are exposed to cocaine in utero and not exposed. So there's a long history of this, but the idea here was that crack is used by minorities, and therefore, accompanying that idea was that crack is more dangerous than cocaine, and therefore those children are more dangerous, more likely to be a biological underclass. So looking at other teratogens, environmental hazards, one is radiation. Higher than normal rate of intellectual disability, greater incidence of leukemia and cancers associated with radiation. And now a new study that just came out, and you can see here, like March 11th, 2021, France grossly underestimated radioactive fallout from an atomic bomb test, study finds. It documents suggests a single test in 1974 called Centaur exposed the entire population of Tahiti, 87,500 people at the time, to radiation fallout. Only 63 Polynesian civilians have ever received compensations for radiation since 1996. Estimated that more than 100,000 people may be contaminated in total with leukemia, lymphoma, and other cancers. So you see here, impact of pollution, literally making these Polynesian civilians, in this case, more susceptible to cancers. So another environmental hazardous pollutant, a longitudinal study found that were from 2001. They were recruited in 2001, I should say, 33,000 of them. Nitrogen dioxide, sulfur dioxide concentrated during 9 months before birth were obtained at the municipal level and assigned to the participants who were born in the corresponding municipality. Responses to survey questions about age-appropriate behaviors at age 2.5 and 5 years as indicators of behavioral development. So the idea basically here is that Japanese officials were able to collect pollution information 9 months prior to the birth of a child. So, during pregnancy, you can find that at the municipal level. And then they compared that to a set of survey questions asked to Japanese mothers at 2.5 and 5.5 years of age. Prenatal exposure to traffic-related air pollution was associated with behavioral developmental milestones, delays of children, and nationally representative sample of Japan. They were more ambitious. They lacked inhibition. They were more impulsive and had a higher inability to compose two-phrase sentences, which was just some test. But the idea here is that pollution during pregnancy was related to outcomes, 5.5 years later. The mother state age. So young mothers are at increased rate of birth complications. So one argument is that the reproductive systems of young mothers are not physically mature enough to deliver a child. Another argument, not necessarily mutually exclusive, is that teenage mothers are obviously going to have more ACEs. They are less likely to seek prenatal care and more likely to be poor. So kind of, again, that cumulative effect of stressors are impacting a young mother's reproductive system. And you can see kind of a chart in the left corner, the different ages. And it's kind of a U shape with young mothers being at risk and older mothers being at risk. So stress during pregnancy. This part HPA axis is highlighted later in the book, but I don't know why they don't introduce it now, so I'm going to introduce it now. So one idea biologically how stress is passed on during pregnancy is through the HPA axis. And the idea here is that it biologically adapts you to stress. So HPA stands for the hypothalamus, the pituitary gland, and the adrenal gland. So the hypothalamus receives a signal for fear and it releases a hormone. And through a chain of biological reactions, eventually the stress hormone cortisol is released. Prolonged and severe maternal stress creates maternal stress hormones would cross the placental barrier and program the fetal physiological system. So the takeaway here is that the HPA axis uh, is a biological mechanism by which stress hormones are released. And during pregnancy, the stress hormones can cross the placental barrier and negatively impact a developing fetus. Now, looking on to natural experiments. So one way to measure the impact of these teratogens, these big stressors during pregnancy, is through natural experiments. So the idea here is a natural disaster, war, global pandemic, and then seeing which parents were exposed to a hurricane, a military occupation, whatever, and then seeing what the impact of that natural experiment, that natural disaster, that war is on a child. In the case of pregnant mothers, you can see the impact of that natural disaster on fetal development. So 300 women exposed to Hurricane Katrina. Pregnant women were interviewed during pregnancy about their experiences during the hurricane. High hurricane exposure was defined as having 3 or more of the 8 severe hurricane experiences, such as feeling one's life was in danger, walking through flood waters, and having a loved one die. The frequency of low birth weight in preterm birth was higher in women with high hurricane exposure than women without high hurricane exposure. So again, the more hurricane exposure, the more your life was in danger, the higher likelihood that you are going to give birth to a child with lower birth weight or a child that's preterm. M23 occupation of Goma DRC, kind of shouting out my own research. This was a Rwandan proxy group that occupied a city within the DRC for 13 days. And I found that mothers pregnant during the occupation gave birth to shorter children. So again, mothers who were exposed to that teratogen, that natural experiment, that military occupation, increased the risk of giving birth to a shorter child. Malnutrition, disruption of formation of spinal cord, fewer brain cells in stillbirth, smaller neurons, smaller brain, smaller child, associated with short and long-term developmental delays. So malnutrition, not good for you. But it is buffered, and I'm going to go into buffers more, but what that means is it's a protective factor. So the idea there's a stressor, and then there's, you have a shield. And that shield, that protection, that buffer can be a parent, a school, a political policy, etc. So the association between short and long-term developmental delays between malnutrition and long-term developmental delays is buffered by parenting practices and school resources. So certain parenting practices and school resources make the impact of malnutrition on development less severe or can completely negate that impact of malnutrition. And the father state, so we talked about mothers a lot, and don't worry about the chart here. I just grabbed this from a random journal article, but the idea is just stress can be passed on. Paternal stress matters. Paternal age, so odds of miscarriage are higher. Elevated risk of congenital heart defects, neural tube defects, and kidney problems, and risk factor for schizophrenia. So the older a father, the more at risk their offspring will be. But paternal stress is hypothesized to intergenerationally pass to offspring through DNA in the sperm. Sperm carries epigenetic information. Female non-stressed rats injected with sperm from stressed rats produce stressed offspring. So the idea is, say, mother's not stressed at all, but in this study, they took sperm from a rat, injected it into the mother, and even though that mother was not stressed, that rat offspring was still showed signs behaviorally of being stressed out. So it was genetically predisposed towards, stress behavior, externalizing problems, whatever it was. And this is exciting new research because so historically, it's been focused on mothers with good reasons, but also we see that these fathers and, increasingly extended households at the family level impact fetal development. So looking at prenatal testing, prenatal detection of abnormalities, we have ultrasound, which is a test in which sound waves are used to examine the fetus. Amniocentesis is a procedure in which a needle is used to withdraw a small amount of amniotic fluid and cells from the sacs around the fetus. And cryonic villus sampling, procedure in which a small sample of cells is taken from the placenta and tested. So the idea is, prenatal testing to see if a child is predisposed for something or at risk for any number of health and genetic negative outcomes. And continuing on with other prenatal testing, maternal blood sampling and pre-implantation genetic diagnosis, just again, more ways to test any potential developmental delays or risks for a fetus. I'm not going to read all these. So maternal illness can impact a developing fetus. So if a mother had a flu, that's generally associated with an increased risk for negative birth outcomes. So this is just kind of a chart of all that. You don't need to memorize every single one of these. The perinatal environment. Childbirth is a 3-stage process. First stage of labor, mothers experience regular contractions. Second stage of labor, delivery. Third stage of labor is delivery of the placenta. Children are increasingly born at home. In 1930, 8 out of 10 births took place at home. In 1990, 1 out of 100 births took place at home. There's a large cross-cultural and historic variations. Why? So, depending on your culture and the historical period, so you can literally see this in 1930 versus 1990. And these, change for a variety of reasons, whatever, the pop culture is of the day, or whatever the new scientific research shows, or, whatever your culture says. It's common in some cultures to give birth at home and others more in the hospital. And possible hazards, brain cells die if starved of oxygen for more than a few minutes. Anoxia, oxygen shortage. So the idea here is, brain cells need oxygen. If a umbilical cord is tangled around an infant, that puts them at risk for long-term cognitive impairments or cerebral palsy. Other possible hazards, cesarean section, a surgical procedure in which an incision is made in a mother's abdominal, has not reduced the population risk of neonatal death. It's estimated that 11% of first-time cesarean sections are unnecessary and 65% of second-time cesareans are unnecessary. Planned C-sections, however, are rated as positive by women who undergo the procedure. And on the right-hand side, you can see the rates of, I gotta adjust this thing here, of the cesarean sections by country. Okay, identifying at-risk newborns. So even though only 9% of births in the US are classified as low birth weight infants, low birth weight infants account for 2/3 of infant deaths. The more lifetime risk factors, chronic stress, the more at risk of pregnancy. So age, class, nutrition. So again, it's a constant theme. The more risk factors, the more chronic stress, mother, father, in this case we're focused on mother, occurs, the more likely or the more risk an infant is to negative developmental outcomes. So one term for this is actually allostatic load, which is a cumulative wear and tear on the body's systems owing to repeated adaptations to stressors, cellular aging, clinical health diagnosis, and telomeres. Summing scores on across a number of biological outcomes. So this is actually kind of similar to ACEs, but it's a biological version. So you just count, do they have high blood pressure? Do they have elevated cholesterol? Do they have C-reactive protein? And you're just kind of tickering it up, and then you're taking each one and then adding it up. The higher the number, the more at risk you are of, or the more evidence there is that your body is kind of has this cumulative wear and tear effect from stress within the environment. So it's a biological way of measuring the impact of stress on an individual. And there's a nice little video if you want to watch that. Other experience, culture, class, historical factors. Different views of desirability of having children. Birth practices vary widely. So in some tribes in Namibia labor by themselves as a show of strength. Women of rural North India have a more hands-on delivery. So again, how mothers deliver their infants has large cross-cultural variation. Class is also correlated with infant mortality. The poorer you are, the more likely you are to have neonatal death. Black infants are twice as high at risk for neonatal deaths than white women's. And postpartum depression, we're going to look at that. 15% of new mothers, previous depression, and lack of social support increases risk. Prolonged depression associated with hyper offspring behavior and elevated cortisol levels. So again, the idea here is postpartum depression. Those mothers who are most at risk for postpartum depression are mothers who were already experienced a ton of other stressors prior to pregnancy. They were already, at risk of depression or depressed prior to pregnancy. So, and then that continues or has a possibility or more likelihood of happening following pregnancy. And prolonged depression impacts the way that mothers interact with their child and impacts the way fathers interact with their child, which then elevates a child's stress level, which is one mechanism by which stress, trauma, depression is passed down intergenerationally. And you can see on the right side the number of births, and you can see that rate going down for millennials, and that was the millennial bullet point. So birth rates going down. They report fear, unprepared, feeling unprepared and helpless. So the idea is that that they have similar stresses as mothers, obviously. So it's the rest of the family. Again, not breaking news. Feelings typically improve. Physiological markers of stress decrease. So you get that initial kind of stress. Oh man, what do I do? What the hell is going on? And then slowly you kind of, hopefully, get used to it, and you're more prepared. You have more practical solutions developed through experience, which decrease stress. And finally, COVID. Messenger RNA vaccines generated immunity in pregnancy and lactating women, similar to that observed in non-pregnant women. So the idea here is that breastfeeding may pass immunity to neonates. And there's no evidence that vaccines affect the women's placentas. And the placentas of 16 women who tested positive for COVID-19 were pregnant showed evidence of injuries. So the idea here is vaccines don't harm a placenta. However, getting COVID does put you at risk for harming a placenta. And that's all for this week. I hope everyone has a great week. Thank you for listening. Bye.